Good evening, all. Am I audible?
Yes, sir, audible.
Yes, sir.
Yes, sir.
Is it visible?
Yes, sir.
Okay. Today, topic for discussion is origin of eukaryotic cell. We all know that eukaryotic cells are the modern form of cells because before that, prokaryotic cells originated from the protocells. Then we got eukaryotic cell in the origin of life form on the earth. So we all know the basic differences between prokaryotic cell and eukaryotic cell. Karyon means nucleus. So based on presence or absence of nucleus, it is divided into prokaryotic or eukaryotic cell. That means the cells having true nucleus are considered as eukaryotic cell. But the cells having primitive nucleus or no true nucleus structure, they are considered as prokaryotic cell. Okay. So in bacterial evolution, we have discussed about the prokaryotic evolution or smaller organism evolution. In this part, we'll discuss about just a minute. Hello. Hello. Ah, it's very class sorry. Good class online class. Okay. Sorry for the disturbance. First of all, we'll discuss about the origin of life on Earth. Then we'll go for the origin of eukaryotic cell. Origin of life. We all know that primitive Earth was completely unfavorable for the life. But due course of time, Earth prepared itself for supporting the life on Earth. Then life originated on the Earth by aggregating certain products present on Earth. Okay. So there are so many hypotheses, so many theories. They suggested about the origin of life, how life came to Earth. Some scientists they told that life originated on Earth by the nature, the God. Okay, that is special creation theory. Not need to discuss here, detailed discussion. Just I am explaining in 1 to 2 lines. Special creation theory explains what? Everything created by God. Okay. In different day, God created different organisms. Okay. After that, some scientists, some educated people at that time, they explained that theory of catastrophism, that life came from outer space as spores. Some people explain that life came from non-cellular components like decaying material, mud. Okay, so all these are the theories they explained, but latter part, these theories were completely eradicated not accepted by any people because there was no such evidences proved those theory. That's why those theory were not accepted by the people at that time, at the latter phase. But when certain theory proved wrong, like theory of spontaneous generation proved wrong by Spallanzani's experiment, by Louis Pasteur's experiment, Swan Neck experiment, everything. The chemical theory of evolution emphasized on origin of life from the non-living component played an important role, which was provided by Oparin and Haldane. What they suggested that life originated from non-living organic molecules, not the non-living components like mud and decaying matter. They suggested that life originated from non-living organic molecules like proteins and nucleic acid structure. That was the that was the analysis of Oparin and Haldane. But when Oparin and Haldane gave their theory, it was not also accepted by many people because Oparin and Haldane, they unable to prove their theory. They only suggested, they only explained their theory by certain assumption, by certain thinking, but they unable to explain this theory due to any factor. Okay. After that, those things were proved by another two scientists, Miller and Urey. We'll go for it. Yesterday also we have discussed at a glance for this Miller and Urey theory. Just here, one picture is there at the right side. How first cell originated. That means when life was formed, before that, certain substances were scattered on the earth's surface. These substances, they do not mean to the life, but after certain time, when those substances get aggregated and in a certain proportion they get aggregated, they form the first cell. Okay, so what was the condition previously was there on the earth and how life originated? We'll discuss about it at a glance. Just look at this picture. This is the condition of primitive life. Okay, heavy rainfall was there, heat was there due to radiation, some UV rays, cosmic rays, all these kind of low intensity light rays are there because there was no protective layer on the earth's surface. Protective layer is the layer of ozone, but that time no ozone layer was formed because free oxygen was not available, which plays an important role in formation of ozone. Lightning was there, severe lightning, vigorous volcanic eruptions were there. Okay, so the condition was like that. The condition was completely reducing condition due to devoid of molecular oxygen, or you can say it as free oxygen. Okay, so what were the substances, simple substances present at that time in this environment? The simple free atoms at its ionic state, they were present in the atmospheric condition like carbon, hydrogen, oxygen, not free oxygen, not molecular oxygen, it is atomic oxygen and nitrogen in its atomic stage. Okay, then after some time, Earth reduced its temperature, and due to all these energies available, that means solar energy or radiant energy, lightning energy, the energy of sparkling, okay, the energy come from volcanic eruptions, all this energy, they help in abiotic synthesis of monomers from simple organic compounds like hydrogen. They form hydrogen atom to molecule. Hydrogen molecule when combined with oxygen atom, they form water. like ammonia and methane were formed at that time. When they combine each other, due to this energy presence at that atmosphere, they form different kind of amino acid, purines, pyrimidines, monosaccharide, fatty acid, glycerol. So this step one, whatever written in this part, this idea was provided by Halden, but they unable to prove their idea. But this idea proved by Miller and Urey. At the latter phase, Miller and Urey, they took the similar components, whatever components described by Oparin and Halden in their flask. Okay, so here already I have explained this thing yesterday. Just at a glance, I am discussing this thing. So in their mentioned ratio, Oparin and Halden, what ratio they mentioned, at that ratio, Miller and Urey, they took that kind of substances what was present in the primitive atmosphere and artificially they created here in this flask, in this glass primitive atmosphere by taking these substances. Okay, along with these substances, that means methane, ammonia, and hydrogen first he took, and along with this he added water vapor by boiling water, and from this side movement of water vapor to this environment. For energy, they provide electrode and they continuously sparkle those electrode for current generation in that environmental situation. That means this is the primitive environment, primitive earth condition, reducing earth condition, and this is the energies available at that time. Okay, so this is actually reducing to oxidizing atmospheric condition. Then what happened? After 17 to 18 days, he marked what substances after condensation, what substances he got in next tube is the substances like organic compounds present in this water. That means water vapor is there. When water vapor get condensed, they form water molecule. But here he incorporated free water molecule. No such substances was present. But what he got here, he got the free water molecule along with certain organic compounds. Okay. So in collection trap, he got particularly certain nucleic acid, fatty acid, polysaccharides, protein-like substances, all these things. Okay, but primitively he got the amino acid, purines, pyrimidines, like this type of substances, and when they got combined, they form all these kind of complex substances like protein, nucleic acid, fat, polysaccharide. Okay, when this protein, nucleic acid, fat, polysaccharide, like this manner, they aggregated and form the membrane. Now, the structure was considered as protocell. And when the protocell developed nucleic acid arrangement in its part, like RNA, okay, and certain proteins are associated with this for the catalytic action, they are considered as fast cell because protocell was not considered as fast cell. Why? It doesn't have the ability, it didn't have the ability of reproduction or cell division. But this fast cell, due to presence of this RNA genetic material, it has the ability for cell division or cell reproduction. Okay, so these are basic idea about origin of life on Earth. So once the first life came into existence, they started evolve in different ways and forms. This led to stepping stone to the theory of evolution. That means when the first cell formed, that was very simple cell. That was not the prokaryotic cell, but prokaryotic cell was the complex cell at that time. From this simple to complex evolution, it took a lot of time. But they change, they adapt new environmental status, they adapt new environmental condition, they accumulated certain products, certain substances, certain organelles inside them, and finally they converted to prokaryotes. So certain theories are there. We'll discuss about it in later. So this all about origin of life on Earth or in the primitive Earth. This is the pictorial presentation. Yes, another important thing is that when Oparin and Haldane explained those things, that means this type of substances, they form the simpler substances, organic substances. The seawater at that time was known as prebiotic soup, where the protocells were formed. First, quaservates were formed, then quaservate developed biomolecules surrounded by a lipid membrane. Then nucleus or nucleic acid originated and protein control the metabolism inside the lipid membrane. That is considered as the protocell, and protocell give rise to a new cell in prebiotic soup. Your classes. Now, come to origin of eukaryotic cell. This eukaryotic cell origin process is known as eukaryogenesis. The process called as eukaryogenesis, by which the eukaryotic cells were evolved from fusion between particularly archaean and bacterian cells. Okay. Yesterday we discussed about the endosymbiotic theory. Today we will discuss detail about this endosymbiotic theory, but at a glance, just when one cell engulf another cell and a symbiotic relationship developed between them in the primitive earth, they converted to eukaryotic cell. Okay. The process is thought to have occurred between 1.8 to 2.7 billion years ago. That means prokaryotic cells were formed before that time. Okay. This is the just pictorial presentation how eukaryotic ancestral eukaryotic cells were formed. In latter part, we'll discuss about this origin in detail. So here certain concepts are there for its origin, how eukaryotic cell originated. The first concept, the well accepted concept or theory for origin of eukaryotic cell was the endosymbiotic theory. What this theory explained, the engulfed bacterium allowed the archaean cell to use oxygen to release energy from nutrients and to protect the bacterium from predators. Okay. Over the time, two organisms develop a symbiotic relationship. Just look at the points, what is written here, what is mentioned here. The engulfed bacterium allowed the archaean cell to use oxygen to release energy from nutrients. That means when the environment converted from reducing to oxidizing, that means free oxygen availability was there. So certain archaean cell, they unable to use oxygen at that time, but the bacterium, they got the ability to use the oxygen for releasing energy. So some archaean cell, they engulf. Look at this ancestral prokaryotic cell, they engulf certain another prokaryotic cell which are heterotrophic in nature. Okay. And they give place those prokaryotic cell into inside the cytoplasm. Okay. When these cells present inside the cytoplasm, now they develop a symbiotic relationship between these two. That means these cells, these prokaryotic cells, smaller heterotrophic prokaryotic cell, they help the whole or host cell in utilizing oxygen. On the other hand, the host cell provide nutrition as well as space to the smaller cell for its survivability. That develops a symbiotic relationship between the two cells. Okay, and these two cells, latter part, they form what? They form eukaryotic cell, ancestral eukaryotic cell. Okay, that's why mitochondria was developed when they engulf the photosynthetic prokaryotic cell. This is normal heterotrophic prokaryotic cell which converted to mitochondria when they engulf photosynthetic prokaryotic cell. The host cell engulf photosynthetic prokaryotic cell. They develop ancestral photosynthetic eukaryote. Okay, so these are different development of eukaryotic cell. The endosymbiotic theory explained about that. Now, inside out model, that means that model suggests some scientists worked on that and they suggested this inside out model. What it suggests, eukaryotes evolved from prokaryotic cell with a single membrane that extended protrusion that fused to form cytoplasm and endomembrane system. Okay, just look at this previous picture, this one. Just look at this picture. Primitively, this is one membrane. Membrane protrusion means certain outgrowths, like this kind of outgrowths. In inward movement, they start protruding, and this protrusion, they develop different organellar structure and nucleus in the eukaryotic cell. That is inside out theory provided by certain scientists. Then the size of eukaryotic cell are typically much larger than prokaryotic cell with a volume about 10,000 times greater. So it is another evidence of this origin, that smaller cells originated first and larger cell after that. Okay. And the diversity of eukaryotes include microscopic single cell organism. That means single cell organisms in certain case, when nucleus and other organelles are fully developed, they are also considered as eukaryote, as well as the multicellular organisms. They are obviously eukaryotic cells. Okay. So this all about the basic idea about origin of eukaryotic cell. And these are the pictorial presentation of this origin. Now come to basic characteristic. All we know this basic characteristic, but for discussion, but for written format, we have to just incorporate this characteristic of eukaryotic cell. First important characteristic is the presence of nucleus. Eukaryotic cell have nucleus which is surrounded by a nuclear membrane. That means membrane bound organelle. It is the most prominent organelle in the cell. In comparison to other organelle, it is prominently visible. Okay. And is often called as true nucleus. In the definition part, from the beginning of the class, I have explained the presence of true nucleus explain the property of eukaryote. And this nucleus contains the cell's genetic material, maybe DNA, RNA, and it control all the activities or most of the activities of cell by direct the synthesis of protein and ribosome. Okay, that's why nucleus is important in eukaryotic cell, and presence of nucleus explains about its eukaryotic nature. Along with nucleus, other organelles are also present in eukaryotic cell. That's why that is different from prokaryotic cell. And those organelles like endoplasmic reticulum, Golgi apparatus, lysosome. This is the picture of eukaryotic cell. Just look at this picture. Whatever organelles present, nucleus, Golgi complex, microtubule, vacuoles, endoplasmic reticulum, centrioles, mitochondria, lysosome, ribosome, plasma membrane, everything. They all are membrane bound organelles. That means all whole cell bears one membrane, the surrounding. Along with the cell, the organelles also bear membrane. That is, they are called as membrane bound organelles. So, presence of membrane bound organelles are another property of this eukaryotic cell, another characteristic of this eukaryotic cell. Now, cytoskeletons, chromosomes are there, locomotions. Eukaryotic cell have flagella and cilia as their locomotory organs, reproduction by mitosis. All these are the basic characteristic. All we know these basic characteristic very well. Not need to explain it in detail. Then these are certain pictorial presentation of first eukaryotic cells. Okay, certain data I got from the internet, and these are the pictorial presentation of those data. The earliest eukaryotic, what is fossil? Fossil is the representation of evidence of those extinct substances. That means from the presence of fossil, we can understand what kind of organism was present previously. Okay, so like that, the primitive eukaryotic cells, that means which cells formed in the primitive environment or long years back, around 1.5 to 1.8 billion years ago, we got this kind of eukaryotic cells like Architrax, Grypania, Spiralis. Okay, so these are basic structures of these eukaryotic cells. These architracts are microscopic organic structure throughout to be eukaryotic microfossil, represent some of the earliest eukaryotic life form. So these fossils actually provide the evidence for the presence of eukaryotic cell or evolution of eukaryotic cell at that proper time. These are the data from the Pangea note. Pangea was the Gondwana land note. Gondwana land was the primitive landmass on the earth. Okay, when life was originated on the earth, that time also Gondwana land was present. Okay, now after origin of this eukaryotic cell, they got diversified. Why they got diversified? Because they are complex in nature. They started division of labor. That means different organelles are there for different function, which is not found in prokaryotic cell. The whole cell is involved in particular function. But here in eukaryotic cell, the basic difference is whole cell is not involved in all the kind of function. Certain part of the cells are involved in certain function. That's why division of labor is found. Based on that, different kinds of cells evolved during course of time, and from three domains of life, first two domains were discovered, eubacteria and archaebacteria. And from this archaebacteria and eubacteria, another group was originated, which is eukarya, okay, or eukaryota. That eukaryota, when formed, they multiply itself. They start its evolution to different form. That's why eukaryotic cells were diversified. The primitive, particularly the primitive eukaryotic cells, were diversified to plants, animals, fungi, various protists, all these groups. Okay. So pictures are at the right side. You can easily visualize those pictures. Early eukaryotic diversification was driven by the acquisition of new metabolic pathway. Okay, so primitively or early eukaryotic cell, the primitive eukaryotic cell, they were completely in the previous structure. Just look at this structure. In this structure, when this kind of cells were developed, this is eukaryotic cell because nucleus were formed, mitochondria was present, but this eukaryotic cell was not photosynthetic eukaryotic cell. When it engulfs the photosynthetic prokaryotic bacteria, then they converted to ancestral photosynthetic eukaryotes. Okay, so this is the condition. Another important property is multicellularity, which arose in several independent lineages, facilitating the evolution of complex organisms. I have already explained that eukaryotic cell, they start diversifying. Just look at this picture. Eukaryotic cells start diversifying from one branch to another because they develop complexity. They develop multicellularity nature. Okay, so this is all about diversification of eukaryotes. Now, major evolutionary transitions. Five evolutionary transitions are there. The basic important concept for these transitions, like symbiosis or symbiogenesis, not symbiosis, that is symbiogenesis, sexual reproduction, and multicellularity. Already we have discussed. That means transition from prokaryotic group to eukaryotic group and from simple eukaryotic group to complex eukaryotic group. There is a great transition. What is the basic cause, major cause behind this kind of transition? The first important cause is symbiogenesis, the origin of mitochondria and chloroplast. Already we have discussed. Picture is at your right side. Second one is sexual reproduction. Why sexual reproduction is important? Because the eukaryotic cell, they maximum time showed sexual reproduction process. In sexual reproduction process, there is chance of allowing more and more complex adaptation due to recombination and other sexual reproduction events. Okay, in sexual reproduction, two organisms, they share their genes, they share their genetic properties. When these two different genetic properties combine with each other, chances of getting complexity is more, chances of getting differences from the previous one or primitive one is more. And multicellularity already we have explained. So this is all about major evolutionary transitions. So this is basic idea regarding eukaryotic origin. Now we'll go for next slide, in which we will discuss about endosymbiotic theory in detail. Just 2 to 2 minutes. Is it visible to all? Am I audible?
Yes, sir.
Okay. Now come to evolution of eukaryotic cell from prokaryotes. How eukaryotes evolved? What is the reason behind it? Now we'll discuss about this. So for this, all the credit goes to Lynn Margulis. She actually proposed the idea and the well accepted idea why there was evolution of eukaryotes from the prokaryotes. Okay. And the theory provided by Margulis, Margulis is endosymbiotic theory. She she proposed this theory in 1960s, and this theory explains how eukaryotic cells evolved from prokaryotic ancestor. So prokaryote are actually the ancestral cell of this eukaryotic cell. It explains that eukaryotic cell originated through a symbiotic relationship between primitive prokaryotic cell. Just look at this picture here. It's a simple diagram. This is a larger cell, this is a smaller cell. When the larger cell, for any kind of purpose, engulf the smaller cell or ingest the smaller cell inside it, the smaller cell, if alive or remain alive inside the host cell, they develop a mutual relationship among them. And that was the theoretical analysis by Lynn Margulis, for which she told that eukaryotic cells were originated because the larger cell do not remove the smaller cell from the body by any means of body process. That love to store the smaller cell without any harm to the smaller cell. They made a symbiotic relationship with the smaller cell. Okay. And in that process, she explained that mitochondria and chloroplast were actually formed inside the eukaryotic cell or complex eukaryotic cell. Okay. What are the steps for this endosymbiosis? This is very important. Just look at it here. First step is the engulfment. When the primitive atmosphere was full with different kinds of cell, larger cell, smaller cell, certain properties bearing cells. Okay. Certain archaea like host cell, mainly according to Margulis, the archaea engulf the bacteria. The larger cell engulf the smaller cell means the archaea engulf the bacteria, who is capable of aerobic respiration. That means why that larger cell engulf the smaller cell? For the food. For the food means there is no survivability of the smaller cell. But if the smaller cell present or reside alive, remain alive in the cell of host, that means host cell allow the smaller cell inside it to survive. And in this process particularly, engulfment occurs for this aerobic respiration. Aerobic respiration means for the production of energy binding molecule, adenosine triphosphate, ATP. And when the cyanobacteria, when the archaebacterium engulf the cyanobacterium were the bacterium which were the property of photosynthesis. They were autotrophic in nature at that time. When those cyanobacterium ingested, engulfed by the archaea, they start developing photosynthesis pigment inside their body, or they start photosynthesis. They start preparing their own food inside their body. So this is engulfment. That means for a particular purpose, the larger cell engulf. Engulf means feeding the smaller one. For any kind of purpose, the larger cell for the nutritional purpose, for any other essential benefits, the larger cell engulf the smaller cell. And then if the smaller cell like aerobic bacterium, cyanobacterium, that means they have certain peculiar and important properties. That was the benefit provided by those cells. And the host cell didn't kill those cells inside their body, because we know that when any other cell ingests into the cell of any other organism, first the host cell try to kill those organisms, because that organism which he was ingested engulfed was the foreign organism, antigenic material. Okay, but here the foreign organism were not killed, were not destroyed by the host, because the hosts start developing a symbiotic relationship with the organism. Instead of digesting the engulfed bacteria, the host cell and the bacteria developed a mutualistic relationship where both benefited. That means the aerobic bacterium provide energy to the cell for its different function, and cyanobacteria help in trapping the sunlight, the alternate source of energy, because the primitive atmosphere, the organisms, the archaean, they use the organic product present in the seawater. But when all the organic product destroyed, they start searching for alternate source of energy, but they didn't have the property of binding the alternate source, that is the sun, solar energy. To bind the solar energy, they didn't have any kind of property. So what they did, they engulfed those cells which have that property, or by any combination, permutation combination method, those cells were engulfed and provide certain services to the larger cell, to the host cell for its survivability. That's why host cell do not digest those cells. Host cell keep those cells inside their environment, and those particular smaller cells, they got protected from other predatory effects. That is the symbiotic relationship. One is getting benefit. That means the larger cell is get benefited by energy, by binding the solar or radiant energy. Okay. And the smaller cell, on the other hand, they got food from the host, they got nutrition from the host, they got protective place from the hosts. Okay, so these are the situations, these are the symbiotic relationship between these two. And the last process of this endosymbiosis is the integration. Integration means over time, many of the genes from the engulfed bacteria were transferred to the host genome, and then bacteria became integral component of the eukaryotic cell. Okay, that means they exchange their material with each other. The ingested cell and the host cell, they mutually exchange the material with each other. And some materials from the ingested cell, engulfed cell, come into the cytoplasm of the host cell, and they remain in the host cell throughout their life. Okay, so this part is known as integration. That means a mutual exchange between these two cells. Okay, so these are the steps in endosymbiosis. Now these are the evidences. So what evidences actually explains? We all know that particularly mitochondria and chloroplasts, we assume that, not we, the scientists particularly, they assume that mitochondria and chloroplasts were the organelles ingested by the cells they converted to eukaryotic cell from the prokaryotic cell for the purpose of respiration and photosynthesis. What was the evidence? We all know that from the previous class, from yesterday's class, we know that bacterial genome is circular, bacterial DNA is circular, and here we can see that mitochondria and chloroplast, they have their own circular DNA, like bacterial DNA. Okay, so this is the evidence. That's why scientists assume these things. What other evidences are there? Both organelles have double membrane with the inner membrane resembling prokaryotic cell membrane. That means they have their own double membrane structure, inner and outer membrane. Okay. They reproduce by binary fission independent of the cell cycle, just like prokaryotic cell. Inside the cell, mitochondria and chloroplasts, how they increase their number? They increase their number by at certain time they did binary fission. Okay, they perform binary fission just to segregate them from one to many. Okay, this is another important property which is independent from the cell cycle. They do not follow the cell cycle. They are the autonomous body for their division. That all the organelles they follow. If we are talking about nucleus, it follow the process of karyokinesis. Other parts of the cell follow the process of cytokinesis as well as karyokinesis, but this mitochondria and chloroplast in animal and plant cell, they do not follow cell division process. They have their own division mechanism like the prokaryotic cell. And ribosomes within the mitochondria. We know that ribosomes were the organelles present in prokaryotic cells, certain prokaryotic cells. Okay. Not the all organelles, but certain organelles were present in prokaryotic cell. Ribosome was one of them. So here, ribosomes within mitochondria and chloroplast are more similar to those found in prokaryotes. That is 70S ribosome. Okay. 50S plus 30S combination of ribosome were found. Prokaryotic ribosomes were found in mitochondria and chloroplast. Whereas the ribosomes of eukaryotic cells, they are 80S, 60S plus 40S Bedburg's unit. Okay. So these all evidence, they explain that particularly these organelles were primitively, the scientists assume that these organelles were primitively present independently outside the cell. During the course of evolution, certain larger cell, they engulf those cells and make a symbiotic relationship. That's why those organs particularly, they function independently from the cell function. They do not depend on the cell function. Okay. So these are the evidences. Now, evolution of eukaryotic genome. So many factors are there. One by one, we will discuss about it. Yesterday, we have discussed about horizontal gene transfer. Here also, horizontal gene transfer is another reason for evolution of eukaryote. Because during early stage of eukaryotic evolution, that means when eukaryotic cells, primitive eukaryotic cells were formed, significant horizontal gene transfer occurred between engulfed prokaryotes and the host. Just look at this picture. This is bacteria like ancestor, endosymbiotic ancestor, okay, organellar DNA. Got it? When they incorporated into the cell, they exchange their genetic material with the cell structure, with the cell or the host cell. The transfer of gene from the endosymbionts, that is mitochondria or chloroplast, to the nucleus played a crucial role in shaping the eukaryotic genome. That means these cells, particularly these, if we are talking about the larger cell, this is the archaea cell, the smaller cells, mitochondria and chloroplast. They primitively from the cyanobacteria structure converted to chloroplast, endosymbiotic bacteria, the bacteria having property of utilizing oxygen, they converted to mitochondria. When they exchange their genetic material to the cell nucleus, there is a combination between this outer cell genetic material and particular host cell genetic material. So this combination give rise or results the property of formation of new eukaryotic genome or evolved form of eukaryotic genome. This process allowed for streamlined function of organelles with many mitochondrial and chloroplast proteins now encoded in the nuclear genome. That means when the properties of both the cells they merge with each other, the cell showing a new property because they start showing new protein synthesis, they start showing new RNA, formation of RNA that leads to another kind of protein synthesis which was not found in the previous cell. Okay, so this recombination leads to evolution of eukaryotic genome after exchange of genetic material within the cells. Okay, genomic complexity is another important reason. When we are talking about primitive eukaryotic genome, they are significantly larger and more complex than those of prokaryotes. Prokaryotic genome are obviously smaller genome because they have small functional ability. When the functional ability become larger, more functional ability found in the organism, the genome structure itself evolved. That's why they contain higher proportion of non-coding DNA, including introns, regulatory sequences, and repetitive elements for different activities. So here you can find certain segments in the primitive part, in the primitive cells. Not so complex or more segments are there. Certain segments were there which can perform certain functions. But when complexity arises, certain segments they united to form particular function. Here one example is given how these codons united to form protein coding region. And this protein coding region, by the process of translation, by transcription, they form the RNA double strand structure, form the single strand structure, and this single strand structure RNA, they when mature, got maturity, they start translation and particular portion is responsible for production of particular protein. This is the region for which genomic complexity was found in eukaryotic cell. Okay. Another important aspect is multiple origins of organelles. That means, like when the mitochondria and chloroplast are the most well-known examples, other organelles may have also originated through similar endosymbiotic events, like secondary and tertiary endosymbiosis in certain algae. We'll discuss about it in later slides. Then gene duplication and divergence is the part of this topic. You should know about it. In yesterday's class, we discussed in detail the gene duplication. Just look at here. What is actually gene duplication? The right side picture explains you the gene duplication, the basic fundamental idea about gene duplication. That means before duplication, the gene structure is this one, the smaller structure. After duplication, that means formation of duplicate, extend the genetic structure. This is the basic idea about gene duplication.
Sir,
So
cause of
what is
gene
actually,
duplication कौन ଅଛି? Cause of gene duplication.
yes,
What is the cause of a gene duplication?
not audible.
Yes, sir.
Any
What is
doubt?
the cause of a gene duplication?
Okay, got your problem. What is the cause of gene duplication? Just think one thing. When environment was not static, environment was dynamic. Okay, organism try to accommodate itself with the changing environment. Yes or no? That is the first arousing cause of this gene duplication. Because gene duplication is actually caused due to mutation, which occur when region of DNA that code for a gene is copied when it is necessary. Got it? Several other causes are there. If we are talking about chromosomal duplication, this can happen when non-disjunction occurs. If we are talking about RNA duplication, then this happens when reverse transcription occurs. If we are talking about unequal crossing over, this happens when homologous chromosomes exchange genetic material. Are you getting my point? Who was asking? Are you getting my point? Okay. So these are different aspects, different circumstances, different causes of gene duplication, which is a major source of genetic innovation and diversity in eukaryotic genome. Particularly, gene duplication explains about the mutation, change in the property, change in the genotypic property of the gene. Got it? Just look at this picture right side. A particular gene at this fragment, duplication happens. That means the particular gene explains the particular characteristics. The homologous gene explain different characteristics. Because just look at this picture. The early globin gene, when duplicated, they form alpha chain and beta chain. And the organisms bear the alpha chain and beta chain or any other kind of genetic property, genetic code. If they get duplicated, organism having that duplicate property is separated from the organism having the parental gene property. And that leads to formation of different species. This is the evolutionary tree formation here. And ortholog paralog condition already we have explained in the previous class, the part of homologous condition. And in ortholog paralog explanation, we assume, we can hypothesize that how that particular gene duplication is responsible for organismal divergence. If genetic divergence are there, genetic variations are there, which is caused due to its duplication, that leads to formation of new species. Because the genetic frequency from the previous species is getting changed. If the genetic frequency is getting changed, then we can say it as this is the process of evolution. Okay. So for that, whole genome duplication, segmental duplication, tandem duplication are basically required. If entire genome duplication or portions of the genome are duplicated or genes are duplicated in sequences are very essential for its change, for its divergence. These are the mechanism of gene duplication. Either whole gene is duplicated or portions of gene are duplicated, or tandem duplication means the particular sequence is duplicated. This is the pictorial presentation in certain duplication format. Whole genome duplication is there, just whole genome duplication. Another pair of genome is formed, duplicated pair of genome is formed. Tandem duplication, particular sequence duplications. Segmental duplication, particular segments. Okay, so these are different kind of duplication pictorial presentations are there, and these duplications are actually responsible for divergence. Already I have explained the property why divergence is responsible for evolution, because genetic frequency is changing, that leads to the process of organic evolution. Okay, and these are the fate. That means what will be the circumstances of duplicated gene. Neofunctionalization means one copy of gene may acquire new function. If frequency is changed, that means certain characteristic may added or certain characteristic may deleted or inverse, any kind of property, mutation property acquired by this gene. So functionalization means both copies of gene split the original function. That means in any duplicated gene, certain kind of actions were altered or changed. Got it? That means addition of characteristic, loss of characteristic, or any alteration, splitting of characteristic observed. These are the fate of duplicated gene. That's why organism subjected to evolution due to change in genetic property. This gene duplication and subsequent divergence enable to evolution of new metabolic pathway, because metabolic pathway by which proteins are formed, proteins mainly control the metabolism of the body, and by this protein, when the proteins are formed, if the proteins are modified by the genes, because protein formations based on translation, which is dependent on transcription. If transcription factors, the DNA which is responsible for transcription is getting changed, that causes the change in translation process, that causes the formation of different protein in the organism. If different proteins were formed, that always responsible for organismal difference from the primitive form of organism. That means if a complex trait in multicellular organism involves the gene families derived from the previous event. Okay, so these all actions, these all duplication properties and different functions associated to this duplicated gene are actually responsible for the evolution. That's why the last paragraph explains the role of gene in the process of evolution. And same thing happened in eukaryotic cell. When eukaryotic cell got their complexity, that means duplication was playing an important role. Duplication is caused due to mutation, which was responsible at that time for the process of evolution. Then now we'll discuss about the impact of endosymbiosis on eukaryotic evolution. Endosymbiosis not only led to the development of key organelles but also accelerated the complexity of eukaryotic cell. Okay, complexity developed because integration of prokaryotic genome as well as archaeal genome. That means the bacterial genome as archaeal and archaeal genome happened that part. The interactions between host cell and its symbiotic partners enable the development of more efficient metabolic process. The organism help the other cell for certain processes. The host allowing the survivability of the bacteria or other cell inside the body. That means the other cell which was ingested primitively is providing certain benefits to the host cell. Okay, aerobic respiration in mitochondria, photosynthesis in chloroplast are actually the benefits, the services provided by the ingested cell to the host cell. That's why endosymbiosis provided eukaryotes with the energy needed to support larger cell sizes, more complex structure, and multicellularity. Okay. So these are certain examples of primary, secondary, and tertiary endosymbiosis. This is the pictorial presentation. Just have a look on this first, then we'll go for the processes. Primary, secondary, and tertiary endosymbiosis. So what primary endosymbiosis explains? When ancestral eukaryotic cell internalized prokaryotes or engulfed prokaryotes, resulting in the formation of chloroplast and mitochondria. The origin of mitochondria and chloroplast form free living bacteria. That is the primary condition. In previous slides, we have discussed the eukaryotic cell and its variation is caused due to gene duplication is one factor. Another factor is that different type of endosymbiosis, primary, secondary, and tertiary. That means in subsequent time, they start engulfing other bacteria having different properties. And those exchange of material lead the assumption for this different kind of divergence in eukaryotic cell. This is the primary endosymbiosis that protist engulfs cyanobacteria. The archaeal group particularly when engulfs cyanobacteria and gene transfer occurs between them. They develop different kind of cell which leads to formation of new branch. Then secondary endosymbiosis explains when one eukaryote like algae is taken up by another eukaryote, resulting in complex chimeras that can have up to 4 genomes. That means we all know that one eukaryotic cell means it has the complexity. If that eukaryotic cell at certain time engulfs other prokaryotic cell or eukaryotic cell, that means they develop more complexity inside them. That thing also assumed to happen in the primitive environment. As scientists explained in different theories, and that is coming under secondary endosymbiosis. Here, tertiary endosymbiosis also there. That means When occurs eukaryotic engulfs another eukaryotic that had already undergone secondary symbiosis. That means after primary symbiosis, secondary symbiosis occurs. Secondary symbiosis organism, if engulfed by any other cells of primary symbiosis, they are called as tertiary symbiosis. That means simple thing, when one cell is engulfing other, the complexity of one cell is merging with the complexity of other cell. When more and more cell engulfed by the primitive cell, they develop more and more complexity. That is the examples of endosymbiosis. These are the pictorial presentation of endosymbiosis. This all about endosymbiotic theory and its explanation. One word is here, which is known as chimeras. What actually the definition of chimera is the imaginary monster. Okay, that means is an illusion of mind, particularly when we'll discuss about chimeras in evolution. It explains actually the single organism composed of cells with more than one distinct genotype. This word is very important. Just look at this word. Complex chimeras means when single genotype allow other genotype to incorporate and they show complete different property from the primitive one. Chimera means actually in the Greek word a single organism having different type of organismal faces. Particularly in certain gods and like that, they use that kind of words chimeras, a single organism having different kind of faces. But here, why complex chimeras are used? When the one symbiotic cell means one cell coming under the symbiotic relationship and got by other cells having the same property, they got more complexity. That means a single genome having a bear more property of expression. That's why that word is mentioned here. So this is all about the evolution of eukaryotes. Now we will go for the next important part, which is the speciation, the first part of the last unit. Today we will discuss about certain parts of this speciation. Then tomorrow we are going to complete that unit. Okay. Just wait for 1 to 2 minutes. I'm just sharing another slide for discussion. So now the word speciation the single word but having a lot of information or containing a lot of information. One by one, we'll discuss about it. So first, we'll go for the definition of speciation. What actually defines it? The process through which new species arise from existing species. This is the process of evolution. When we are talking about new species, that means evolution already happened. Otherwise, we cannot talk about new species. So, it is the process by which new species or population evolved to become distinct species. Distinct species means which was not present primitively in that population. That is called as speciation. There are several mode of speciations are there. Okay, which based on different kinds of isolations. So four important speciation property we'll discuss about. We are going to discuss allopatric speciation, sympatric speciation, parapatric speciation, and peripatric speciation. So it is very important and interesting concept. Listen this carefully. First of all, we'll go for this allopatric speciation. Just look at the interesting diagram present at right side. What allopatric speciation actually explains? Allopatric speciation, known as the geographic speciation. That means if geographically organisms are separated from each other, geographic position means the habitat separation is there. Complete habitat separation due to any factor, any environmental factor, if habitat separation is there, then the speciation is called as allopatric speciation. Is the type of speciation that occur when a group of same species become isolated from the rest of the population. This isolation can be caused by physical barrier like desert or river or by great distance. This is original population. Okay, when there is geographical barriers are there, that means original population due to any kind of environmental, if there is tectonic plate movement, if there is any other destruction due to desert formation, okay, that means they got separated from each other. And if the separation, the originality maintained due to sexual relationship between these two species group, but when they got separated, they try to adopt themselves in new environment, and their adaptability in new environment develop the new characteristic, and that lead to formation of new species, that is known as allopatric speciation. So here allopatric speciation is caused basically due to what? Due to geographic isolation. This is important property of allopatric speciation. Here are some characteristic I have cited. Just look at this characteristic. Isolation, basic physical barrier. Isolation means it's a physical barrier or great distance prevent the group from mating with the rest of the population. Just look at this population diagrammatic presentation here. Here the population was the original population. That means all the kind of species are present in that population. The population has its own identity when there was no separating force. Geographic separation or any other kind of separation force was acting on that population. The population had its own originality, identity. But when isolation occurs, that means the particular group of population may be homozygous, dominant, heterozygous, homozygous recessive, every kind of genomic uh, organisms, genotypic organisms present in that population, when they got separated from each other due to any kind of geographical barrier, they got isolated from each other. That means this geographical barrier, time after time, do not allow the organism here between all the organism chance of mating, which is called as random mating, was possible. But due course of time, random mating means every organism has equal opportunity of mating with other organisms present in that population. But here, random mating was completely prohibited due to this factor. This geographic isolation leads to mating or prohibition of the mating between these kind of species. Now they start developing their own property in different environment. If geographical isolations are there, they separate from each other and they start adapt themselves in the new environment. Just look at this picture. The upper one developed certain characteristic and the lower one was similar to the primitive characteristic, the original population characteristic. The upper one, due to its movement to, might be movement to another area, and the area was not similar to the primitive area of the primitive population. That's why they developed certain characteristics, and those characteristics help in formation of new group of species based on that particular characteristic. Okay, which is completely isolated, which is completely different from the original group of population, and that leads to the last characteristic of allopatric speciation, which is called as evolution. So this is the explanation of allopatric speciation and its characteristics. So many examples I have cited here, 2 examples. You can cite so many other examples by from other sources. Okay. So this all about allopatric speciation, particularly it explains about geographical isolation. That is the first speciation. Now come to the second speciation, that is sympatric speciation. Sympatric speciation is another important. That means evolution depends on this speciation, these factors. Okay, sympatric speciation means new species evolve from single ancestor. That's why that is sympatric. New species evolve or evolve from single ancestor while living in same geographical time in the same geographical area. That is the sympatric speciation. It's a process in evolutionary biology that describes the evolution of new species from a single ancestral species while both the species continue to live in the same geographical area. That means in the previous one, due to geographical isolation, the species unable to mate with each other. Sexual reproduction was not possible due to geographical isolation because the isolating cause is so powerful, organism unable to cross that and meet other organism. But here another factor act on this. For this factor, they unable to mate with each other, though they are sharing the same habitat, and that leads to the evolution of new characteristic. That means formation of new species. Okay, so this process can occur when reproductive isolation prevents in production of viable offspring, which produces gene flow and leads to genetic divergence. Okay, here some terms are there. You have to understand those terms first. Sympatric refers to organisms that share a range and can be found together in some places where sympatric speciation is the process by which new species evolve from single ancestral species. Okay, so here one word is there which is called as sympatry. Sympatry, how we explain sympatry in very easy manner? When two or more species of population live in the same geographical area and frequently encounter with each other, that explains about sympatry. Okay, those species are called as sympatric species. But when these species stop meeting frequently, these species develop certain characteristics which do not allow other species to mate in the same geographical area, that leads to sympatric speciation. Okay. So one example, if we'll discuss, we can understand it very well. If we're talking about certain apple maggot flies, the flies originally laid their eggs in certain places. Hawthorne was the place, the suitable condition for egg laying of these flies. But after apple was introduced to North America in particularly 19th century, some began to laying their eggs in apples instead. The two types flies are not yet separate species, but many scientists believe they are undergoing sympatric speciation. That means due to particular characteristic, due to particular nature, they do not interbreed with each other. Okay, that means particular. If we are talking about the cichlid fish develop in the same geographical area, and those fish develop certain characteristic like coloration due to certain environmental factor, their original population stop mating with those new population group, and but those new population group are actually not the different population group. Due to certain environmental factor, they develop certain albino type characteristic, but those characteristics are actually the determining factor for different speciation. Particular those albino species, they got met with each other, but not the original species. Though they are sharing the same habitat, they do not met with each other. That is the basic idea regarding sympatric speciation. Though they are sharing the same place, the organisms, they are sharing the same place, but they start developing different characteristic by chance, and those different characteristics do not allow the original population to mate with this population, and which make a complete barrier between these two. The two now become different diversified group of population. This phenomenon is known as sympatric phenomenon. No geographical isolation, nothing happened, but sexual isolation happened due to this type of characteristic. Okay, so this is all about sympatric speciation found in organismal group. Now we'll discuss about peripatric speciation. One speciation is known as parapatric speciation. Another speciation is known as peripatric. So first of all, we'll discuss about peripatric speciation. what it explains? It actually explains where a new species is formed from a small isolated population that break up from a larger population. Okay, already this idea I have given, I have provided in previous class, in yesterday's class. Just remember the thing, the genetic drift, the concept of founder effect, the concept of bottleneck effect, but particularly founder effect is the important concept here. Okay, so when this peripatric speciation form, it formed when new species form from a small group of species of original population or isolated population, which are considered as founder population. Okay, when a small group of individual become isolated like this, just look at this picture. This is the original population. Small group of original population due to any factor, due to predatory factor, due to reproductive factor, due to any other kind of survival factor, they just separated from their original population and they start residing at a new environment from the original population. That is the isolated population. That is the small isolated population. Okay. And they develop certain characteristic in the new environment because those characteristic was not found. If we are talking about the isolation of small group of Darwin's finches, whatever finches are present in primitive island or the mainland, they have certain or fixed big structure. But when a group of finches, small group of finches, due to any factor, they migrated to the island and unable to return back to the mainland, they try to survive themselves. They struggle for their existence in the new island and they survived. That's why they are called as founder organism of that new island. And they start developing certain kind of organism of their own kind, which were completely different from the organism in the previous time. That kind of speciation is known as peripatric speciation. That means a small group detached from the original group and they develop a new kind of population. Just look at this picture. The green is the small group which separated from the original group and they converted to blue. That means they developed certain new characteristic in the new environment for adaptability. Due to development of this characteristic, they develop adaptability nature. Okay, this is known as speciation because new species, new characteristics are formed, and this kind of speciation is known as peripatric speciation. Example here cited fruit flies example you can cite the examples of Darwin's finches. Similar to allopatric speciation, the peripatric speciation is almost similar to allopatric speciation. What we have discussed in allopatric speciation, a group is separated. Here also a group is separated. But in allopatric speciation, geographical isolation is one of the important cause. But here, there is no need of this geographical isolation. Organism can move from its own choice. That is also another cause. That is also one of the cause for this type of isolation. Founder effect explains what? When the new group try to survive, struggle for the existence, and they got success in surviving in the new environment, they are called as founder organism. Okay, and those founder organisms are responsible for speciation or formation of new group of organisms. That is the peripatric speciation. Now we'll go for the last kind of speciation, that is parapatric speciation.
Hello, sir. Good evening.
Not audible? Any doubt?
Yes, sir. Good evening, sir.
Okay.
Hello.
Parapatric speciation, what explains to us? It particularly
Sir, I
explains.
have a doubt.
Anybody is saying anything?
Yes, sir. I have a doubt.
Yes.
Sir, actually, after isolation, is the new species is totally different from the main species?
Again, in isolation,
In isolation, after isolation in peripatric speciation,
yes.
is the new species is totally different from the main species?
No, I'm not talking about total difference. Here, speciation means same group of organism is different from the original group. That means evolution doesn't mean that a complete new organism formation. Evolution means any of the characteristic that may be a minute genomic characteristic it changed, and which is responsible for certain that might be smaller or larger change in characteristic of the new group. That means if a particular sequence of amino acid or genetic code is changed, it might not be the reason for the change in whole organismal structure, but it helps in changing certain properties of the organism. Think about the beaks of Darwin's finches. Finches were almost similar to the mainland finches. But what Darwin marked at that time? Darwin marked particular difference at the beak shape, and those beak shape and structure as the responsible cause for their survival in the changing environment. Changing environment means those island environment. That leads to the process of evolution. Okay, are you getting my point now?
Yes, sir.
Now come to the last speciation, that is parapatric speciation. What it explains? It explains a type of speciation where two subpopulations of species evolve reproductive isolation while still exchanging gene. That means gene flow is taking an important role, or it has important contribution. Okay, where when it occurs, when parapatric speciation occurs, it occurs when population expand into a new environment, or that tries to incorporate certain group of population tries to incorporate or integrate into another group. Expansion means this is the factor of expansion. Adjacent population evolve into distinct species while maintaining contact along a common border. That is the meaning of parapatric speciation. Just look at this picture. This is one population, this is another population. But when this population try to expand themselves means try to expand themselves in this direction, or this population try to expand in this direction. That means a hybrid zone is created here. This is very important. Hybrid zone explains the development of new characteristic. That means certain fixed characteristic for this population. Here for this population, another fixed characteristics are there. But when these two population group come in contact with each other, that means they develop mating characteristic, reproductive exchange, and this particular zone Due to recombination, due to sexual reproduction, they develop certain new properties inside them. They have properties of these primitive organisms, but due to intermixing, they develop certain new properties, which which actually responsible for evolution. Okay, so these are the characteristic of parapatric evolution. Isolation, that means parapatric speciation occurs when population is isolated, usually at the edge of a larger group by extreme change in habitat. When this group expand themselves to this direction, they come in contact with this group and they form a new hybrid group. The new color was developed here. That means new hybrid group was developed. Hybrid means combination of these two. Now, this hybrid group, they do not adjust themselves because these two groups, they do not allow themselves to stay with them. They feel separated, isolated from their original population group due to change in nature. That is the isolation. Gene flow may continue between two population during speciation process. Okay. Hybrid zones already I have explained. Reproductive isolation, that means when these organisms, they do not feel free in this zone, or they do not feel free to mate with the original population. That means they start developing reproductive isolation category. They do not mate with each other. Reproductive isolation means stop mating with each other. Stop mating with previous population, parental population. Okay. So this all about parapatric speciation. When if we are talking about the species like certain flowers or grass species here cited, you can find the parapatric speciation. Okay, the picture here, the parapatric speciation means new niche is developed due to incorporation of other group. Okay, and there is a hybrid zone. This hybrid zone is separate from this original or primitive groups. The speciation occurs, the formation of new species occurs, and this kind is known as parapatric speciation kind. This is all about the four types of speciation, what we have cited in the first slide of speciation. Got it? Here, sympatric, allopatric, and parapatric mentioned in the right side diagram because sometimes the allopatric is explained in the form of peripatric group. Okay, but already we have discussed all the kinds in its individual format. So all these are the explanation and certain examples of different kind of speciations. Now we'll go for the factors which are responsible for speciation. One by one, so many factors are there. One by one, we'll discuss about those factors. Several factors, that means the causes of the speciation. Several factors are there which drive speciation. That means they are responsible for speciation. Actually, they influence the genetic divergence and reproductive isolation. Reproductive isolation is actually the cause for speciation. If one group is separated from the same parental group, that means the particular separating group is unable to reproduce, unable to mate with the parental group. That is the reason for their separation. Okay, so geographical isolation is the first important cause for this kind of separation. Just look at this picture. The two groups were actually primitively not two groups, they were one group. But when geographical isolation occurs, a river is the isolating cause in between these two habitat. Then these two habitat is different from each other. Now, the original group, just look at their morphological structure. They were almost similar, but they stopped mating with each other like previous form. Due to isolation, they stopped mating with each other and they develop different characteristics in new environment. Some of them, they bear the original characteristic, okay, and some of them, they completely different new characteristic in new environment. And when they met each other after a long period of time, they unable to meet with each other, which is responsible for their reproductive isolation. So geographical isolation is the cause for reproductive isolation. Now come to reproductive isolation. That's the mechanism that prevent inbreeding. Inbreeding means mating. Same organismal breeding is inbreeding, mating. Mating is possible within same group, not possible within different group. So inbreeding between diverging population, if occurs, that is known as reproductive isolation. This reproductive isolation is again divided into pre-zygotic barrier or post-zygotic barrier. Pre-zygotic barrier means before zygote formation, the gametes, if no copulation, if no mating, then organism unable to form the gamete. Okay, that is known as pre-zygotic. Here, just look at this two picture. This is the zygote. If sperm and ovum, they do not mate with each other, they do not come in contact with each other, that means this cross mark is appropriate. That means there is no formation of this zygote. Okay, how this thing happen? Due to geographic isolation or due to any other factor? What is post-zygotic? Post-zygotic isolation or barrier is due to reduce the viability or reproductive success of offspring. That means if there is mating, but organism develop its sterility, organism unable to form the hybrid, unable to accommodate the hybrid, then that concept is known as post-zygotic reproductive isolation. That means formation of new species is not possible. Though zygote is formed, mating is there, but due to any circumstances, due to any activity, due to any factor like sterility, hybrid breakdown, in so many organisms, we can see that the organism unable to maintain its hybrid. Okay, that is known as hybrid breakdown. Hybrid means two organisms come in contact, mate with each other to form a new organism. That is the hybrid. But when these organisms unable to form the new organism, though zygote is formed, that is known as post-zygotic barrier. So pre-zygotic barrier as well as post-zygotic barrier, they are responsible for this reproductive isolation. So these are two important factors responsible for speciation, geographical isolation and reproductive isolation. So many other factors are also there. Not only these two, so many other associated factors are also responsible for this isolation. So here, one of the important factor, another associated factor is ecological factor. Ecological factor is the divergence in habitat use, feeding strategies, niche specialization can lead to speciation. Just look at this picture, the first picture, this picture. When we are talking about this, these are the main island. When the finches move to this, finches move to this, finches move to this, these are groups of island nearer to the main island, which are having different type of habitat specialization. That means in certain habitat, the feeding components are different. In certain habitat, the habitat space is different. Habitat climatic conditions are different. Okay. So in different circumstances, organisms, when moved from the main island and unable to return the main island, some of the organisms try to survive themselves. That's why they tried survival. They struggle for their survivability. Okay. This is the ecological factor which is responsible for speciation. This is another important factor. And rest all factor from this we'll discuss about in next class. Okay. So for today, this much. I'm just stopping the sharing. Yes, Jyoti Mahapatra raised her hand. Any question?
Sorry, sir, I have raised it first before asking the question,
Okay.
and I have no
So for today,
doubts now.
so this much for today. I'm going to stop. I'm going to end this meeting. Have a good night to all and thank you.
Thank you so much, sir.
Thank you, sir, so much.
Thank you,
Very
sir.
good night.
Thank you so much.